Good. So welcome back to Monday night, biology night. It's great to see you here. It's Julia from STEM Zone. And every Monday we talk about different concepts in biology. And in particular, at the moment, we're focusing on Unit 3 for Year 12s. So tonight is a quick tutorial on cellular respiration. So you guys have been talking about biochemical pathways over the last few weeks. You've talked about photosynthesis and that amazing reaction. And now you are looking at that second pathway that BCAA have said, this one's important. Make sure you have a look into this reaction, and that is cellular respiration. So I'm just sharing my screen and sort of getting sorted here. Always like to share our screen on these lives so that you can just see images and go along with us. And when you watch this back later, it'll be there. You can pause it, have a look at diagrams, etc. So is that good? Right. Look at this amazing diagram. So this is a digital representation of a particular organelle. And all through like junior high school, you guys talked about something being the powerhouse of the cell. So as a Year 12 student now, you guys are very much delving into the detail that you've never sort of learnt before. So this is an image of a mitochondria, and every different coloured piece there is a different sort of molecule found within the cell and found within the mitochondria there. It's a big representation of the complexity of all the reactions happening in a cell. So you can see that mitochondria, it has an outer membrane, and then this beautiful folding cristae, that inner membrane there going throughout the mitochondria. So I put this photo, this sort of image up just to engage with you to start with, to think about, you know, when you guys are learning about photosynthesis and cellular respiration, you see sort of it as this 2D kind of chemical reaction, an equation on a page, right? You're not actually thinking about it in real time. So this reaction that we're going to talk about today, we're just going to go through the 3 steps of cellular respiration, the 3 main steps. This reaction is actually the reason that we breathe. This is directly linked to why you breathe as an individual, as a multicellular organism, which is mind-blowing that it all comes down to this chemical pathway. So let's just get started. There's 3 stages to this amazing reaction. You've got glycolysis that's happening first. The first glucose molecule in action is split in the cytosol of the cell. So glycolysis, glycolysis, is a splitting of glucose into 2 3-carbon molecules known as pyruvate. So glucose is a 6-carbon molecule. When you split it in half, you're left with 3 carbon molecules. We often follow the carbons through these molecules as well. When you talk about DNA, you've looked at how many carbons there, and you've got the 5' and the 3' end of DNA based on the carbon numbered in the sugar. So we're looking at carbon here. In the cytosol of the cell, you've got glucose split. What you get from that reaction is 2 pyruvate, but you also get the creation of 2 ATP, 2 net ATP molecules, so energy there, and you get a bit of NADH formed as well. And the BCAA says it's really important that you guys understand how many ATP molecules are made each stage of the way, because that is the ultimate goal of this reaction. We can consume all the glucose we like, and glucose is a high energy molecule, but it's all stored up in the bonds in glucose, and it's not usable energy. So we want to break down glucose and go through the stages to be able to generate ATP from that glucose. So ATP, adenosine triphosphate, is that usable energy that we need. This is why we do cellular respiration, right? So we've got glycolysis happening in the cytosol. If there's oxygen in the environment, then it will proceed to the further stages, and this is known as aerobic cellular respiration. So we've got glycolysis occurring in the cytosol. That pyruvate will then, you can see up the top there, go from the cytosol into the mitochondria. And so the beautiful purple space is the mitochondrion. Now, pyruvate is oxidized and loses this carbon, which forms carbon dioxide, and that's released. So pyruvate was a 3-carbon molecule. It's undergoing a process where it's transformed into acetyl by losing that carbon, and coenzyme A comes on board. So together, they form a new molecule called acetyl-CoA. Again, in biology, we're not too fussed on all of the chemistry details there, but you do need to know that pyruvate is just the first sort of step. That's the product of glycolysis, but that can't enter the Krebs cycle or the citric acid cycle, the next part of cellular respiration. So it needs to be processed, and we've got a coenzyme added there. So acetyl-CoA or coenzyme A is formed, and in doing so, we also make some more NADH. So we've got NADH made in glycolysis, and then again in this link reaction or pyruvate processing. And you've got your first carbon dioxide release there. So acetyl-CoA, so ultimately our journey for glucose is that it's slowly being destroyed and broken down. And the products made along the way in that sort of breakdown are used in the final step to make that usable energy. So we're looking at this, we've lost already a carbon from the 3-carbon molecule. Now we're in acetyl-CoA, we've got 2 left. And it undergoes the Krebs cycle, which is a series of enzyme-controlled reactions where it separates that acetyl-CoA out. In doing that, can you imagine breaking the bonds, breaking apart this, what was once glucose, right? You're releasing electrons, you're releasing hydrogen, you're releasing carbon. And so there are electron acceptors there. There are coenzymes, unloaded coenzymes waiting around, NAD, NAD plus, and FAD, FAD. So our friends NAD and FAD are waiting there near the Krebs to collect those electrons. And when you collect electrons, they're negatively charged. So that means that you become more negatively charged. You are negatively charged. And obviously then you would attract some hydrogen, some positive ions there. Now, guys, I'm talking like you know all about this, but we did talk about this first in photosynthesis. So it's the same kind of idea. These guys pick up electrons, they pick up hydrogen, they become loaded electron carriers or loaded coenzymes, and they're going to make their way to the next and final stage. So really important there that you understand the outputs of the Krebs cycle. We've got NAD and FAD, our friends, picking up electrons and hydrogen. We've got an output of CO2, carbon dioxide, as we break apart that acetyl coenzyme A, that breaks apart, it's released as carbon dioxide. And you also get a little bit more ATP made. So an input for the Krebs cycle would be adenosine diphosphate and inorganic phosphate, PI. And an output would be ATP, adenosine triphosphate. And you get 2 ATP, net ATP in this phase of the reaction. So far, we've got glycolysis that gave us 2 ATP. We've now undergone the Krebs cycle, which is 2 ATP. So that's only 4 little molecules of usable energy. The ultimate goal is to get the maximum amount of usable energy from one glucose molecule. So that ends up being, if possible, if it goes through the whole series of aerobic respiration reactions, then it can be up to 30 or 32 ATP molecules at the end. And that's the goal. If you just got a measly 4 and had to stop for some reason, if you just did glycolysis and just got 2 ATP molecules from one glucose molecule, what a shame that would be when you can actually obtain up to 30 or 32, right? So this the goal of this reaction is to get that ATP for the cell to use it. All right, so I just want to, just before I move on, talk about our friends, NAD and FAD. And when I say our friends, we met NAD back in glycolysis. So NAD was the first friend we met, NADH. You'll notice that when it's loaded, when it's Sorry, guys. When it's loaded, NADH, it reminds us of another molecule. It reminds us of NADPH, which was the electron carrier from photosynthesis. It was the coenzyme from photosynthesis. So we know the difference between NADH and NADPH because the P is for photosynthesis in my mind. And that's really helpful and serendipitous that it has a P there. So NAD is our first friend that we meet, and it's NADH, is one H, one hydrogen. So actually I've got something missing here. I've got a little positive sign missing. It should be NAD plus. Because NAD plus will pick up 2 electrons. If it picks up 1 electron, it becomes neutral. Picks up a second one, it becomes negatively charged, 1 negative. And so then if it's negatively charged, it attracts the hydrogen and becomes NADH. I won't go through that detail too much. Talk to your teacher about it if you really want to understand, but it's just important to remember that we need to name these molecules correctly. So it's NADH for NAD. For FAD, though, it's FADH2. That name is talking about 2 hydrogens there. So the way that I remember that one, FAD is not FAD plus, it's just a neutral molecule to start with. And so when it picks up the 2 electrons, it has a -2 charge. So to become neutral as a molecule, it will attract the 2 hydrogen and end up as FADH2. And so when I talk about these guys, I talk about NAD and FAD. And FAD's my second friend. And it has 2 hydrogen. That's how I remember it. So NAD was the first one we were introduced to in glycolysis. And then NAD and FAD are both created here in the Krebs cycle. FAD's the second friend. FAD has a 2 there, FADH2. Just remember that important one, because if you write it incorrectly, you don't want to lose a mark just because you didn't name it. So far I've got 4 ATP. I have finished with the Krebs cycle and I've made my products NAD and FAD and ATP. ATP is going to be used. NAD and FAD are needed for the last reaction. Last reaction might look a little bit familiar. It's the electron transport chain, which is very, very similar to the idea of the electron transport chain in photosynthesis in the light-dependent reaction. But there are some key differences there. So here's our electron transport chain. This is part of the membrane of the cristae. Here you've got the cytoplasm up the top in light green. So you can see this is the membrane, the sort of outer membrane of the mitochondria, that first layer. Then you've got the intermembrane space, and then you've got your cristae membrane, so your inner membrane. Then inside that area is the matrix. One of the best series of movies ever. Cool. So you have these proteins embedded in the membrane that transport electrons. So it's called electron transport chain. NAD and FAD, your friends NAD and FAD are needed to drop off electrons to drive the electron transport chain. In the photosynthesis electron transport chain, remember you had water splitting to form oxygen and hydrogen, and it was feeding the chlorophyll with those electrons from splitting water, and those electrons were going through the chain. Well, in this scenario, there's no water being split, there's no light exciting the electrons. The electrons are brought by NAD and FAD. And as they drop off the electrons, they're also losing their hydrogen. So hydrogen builds up as a concentration in the matrix there. But as the electrons go through the chain, they power these proton pumps to push hydrogen across the membrane against the concentration gradient. So this is a higher concentration of hydrogen up here, and we've got our sort of lower concentration here, but we want to fill this space with hydrogen. We want a really big difference. We want a strong gradient there. So electrons go through and they power the pumping of hydrogen into that space. Now, as the electrons go through the chain, they do need somewhere to go. Electrons are not just going to hang around and fill up these spaces, and like they're going to have a major effect on those molecules if you've got electrons, extra electrons everywhere, and they would just bank up like a traffic jam. So there has to be something there to take those electrons away. They're going through the chain, through the transport chain, and then you've got oxygen here as the final electron acceptor. It's there waiting, not waiting. It's around the area and it will attract those electrons. When oxygen grabs those electrons, it becomes negatively charged and attracts hydrogen that's in the area. Hydrogen, remember, came from Nat and Fad. Hydrogen's in the area and the oxygen picks up those electrons and attracts hydrogen, become water. So one of your outputs for the electron transport chain is water. Guys, where do you get oxygen from? This is why you breathe. Have a think about it for a second. You have developed, as a multicellular organism, we have these extraordinarily detailed, finely tuned breathing apparatus in our body to maximize the amount of oxygen that you can feed into your bloodstream to circulate to all of your cells. Just so we can get oxygen, it seems, so that it can be present to accept electrons in the electron transport chain in your mitochondria, to maximize the amount of energy you can get out of glucose and make lots of ATP. This is why you breathe. Okay, so oxygen is the final electron acceptor, and in doing so, it forms water. But the main thing here that we're interested in is the fact that hydrogen is in a higher concentration on the inner membrane space. So for hydrogen to move, like molecules, you know, and atoms and things, they move towards equilibrium. They sort of spread out until they're evenly spread out. But hydrogen ions can't just cross the membrane through the phospholipid section because they're charged. They're a charged ion. They can't just go through lipids. The only way back through to reach equilibrium is through ATP synthase, a protein embedded in the membrane there. So hydrogen's building up as a high concentration up here. Each hydrogen then that goes through ATP synthase powers this enzyme to put adenosine diphosphate and inorganic phosphate together to make ATP. So every time a hydrogen goes through, that's the only way hydrogen is able to get to the other side towards equilibrium, equal concentration. Every time a hydrogen goes through, it powers ATP synthase to make ATP. And what was the goal of cellular respiration was to break down this glucose through this series of steps and to finally produce usable energy. And along the way, you made a little bit of ATP in glycolysis. You made 2 ATP. Then in the Krebs, you made 2 ATP. And now in this final stage of the electron transport chain, you have made 26 or 28 ATP. You really want to get to this stage because this is maximizing the amount of usable energy from glucose. So, guys, if there's no oxygen around, oxygen cannot be the final electron acceptor. So we do get this halts in the system and you don't get this ATP made. So instead what you'll get is in anaerobic conditions where there's no oxygen around or little oxygen around, you will get glycolysis occurring, absolutely, in the cytoplasm. You'll get glucose split, glucose split, glucose split. You'll get those 2 ATP for every glucose, but you won't get the maximum amount here, as in aerobic. So that is in a nutshell. We do love our diagrams with biology, and so sometimes it helps to summarize what goes in, what comes out, sort of thing. But you've got to make sure that you draw it in a way that makes sense to you. So in this diagram, for example, outside of that nice purple mitochondrion, outside here in the cytosol, we've got glycolysis occurring. Glucose is split into 2 pyruvate. In doing that, we end up with 2 ATP from that, a net of 2, because actually I think 4 are produced, but we use 2 in the process. So there's an output of 2, plus we meet our friend NAD. If there is oxygen around, pyruvate will then undergo oxidation and form acetyl coenzyme A, or acetyl-CoA. Part of that process releases some more NAD. So NAD's a friend along the whole journey. Acetyl-CoA can then also enter now the Krebs cycle, where it's just further broken down. That cycle gives you a little bit more ATP. And it gives you more NAD. And we meet FAD, our second friend. So NAD and FAD are made in the Krebs, and FAD is FADH2, our second friend. So both of those guys are needed to transport electrons and hydrogen to the electron transport chain, where oxygen is there to accept those electrons, to keep the chain moving. And what we get is 26 or 28 heaps of ATP in that final stage. I did say this is why we breathe. I said oxygen is literally waiting, is there to, you know, accept these electrons and be part of the system to get energy. The other reason, though, you breathe is because as you're breaking down the glucose, you're releasing the oxygen as carbon dioxide. And as that builds up in your blood, carbon dioxide becomes sort of toxic. It makes your blood more acidic. And so you're breathing out all of that excess carbon dioxide made. You're breathing out that waste so it doesn't accumulate in your blood. And that's it. Some people resonate with a table. This is a bit too much information for me. Just remember, though, that VCAA have said, please remember the number of ATP of each stage, but only if it suits you. You can delve into the numbers of all the other molecules and things like that. Don't be too stressed about that if it's a bit challenging. So just tell yourselves a story, though, of each stage, because it's a story that will help you remember what does go in and what does come out. And that's it for today. I'm going to stop sharing. Hopefully that was a bit helpful. But go around and ask everyone, ask people at home, why do you even breathe? Why do we even breathe? Because oxygen is the final electron acceptor. And we need to get rid of that carbon dioxide so we're not acidic. All right, I'll leave it there. It was a bit of a long one. Thanks, guys, for tuning in. And Carissa will be back tomorrow. Love a good chemistry and psychology tutorial as well. So we'll see you then. And be in touch if you got any questions about anything. See ya.